So before pressing on, there was a question that a few students had about the definition of diffusion. So we're going to go ahead and define diffusion a little bit more. And the reason for this is that diffusion, if you look up diffusion, if you Google it, what you'll find is the definition of solutes moving down their concentration gradient from an area of higher concentration to an area of lower concentration. Now, that is diffusion, right? But that definition of diffusion is assuming that the membrane potential is zero. So if the membrane potential is zero, definition of diffusion just becomes going from an area of high concentration to an area of low concentration, membrane potential being zero. But this definition fails when the membrane potential is not zero, because then you have to take into factor both the potential gradient and the concentration gradient, right? So if the membrane potential is some number x, right, then you have to not only worry about the concentration gradient, but also the potential gradient. Now, if the molecule does not have a full charge on it, then the potential gradient goes away, and it's like our definition becomes the normal definition of going from high to low concentration, right? But if the molecule does have a full charge, plus or minus, then you cannot ignore the potential gradient. And in the case of ions, well, you cannot ignore the potential gradient. And this is, of course, if you have a potential on the membrane. So in the case of 140, biology 140, when you're taught about diffusion, is that, yes, molecules move from higher concentration to lower concentration, but we never had, at least when I teach it, I don't talk about the electrochemical gradient, because I never talk about the potential on the membrane. So when I teach diffusion, I'm talking about things moving from higher to lower concentration, and the membrane that they're crossing has no potential on it. Now, what these two things have in common, when you have a potential and you don't have a potential, is this movement. The reason why it's called diffusion is because the movement will have a delta G less than zero, right? And this one will have a delta G less than zero, meaning that in both cases, the movement will happen by itself and not need to be coupled to another favorable reaction for it to occur. In both cases, the delta G is less than zero. So the real definition of diffusion is the movement of molecular ions from one place to another, where that process is going to happen spontaneously, meaning that it has a delta G less than zero. And that covers every scenario, meaning that, well, if you don't have a potential on the membrane, well, then you're just moving from higher concentration to lower concentration, and this is going to happen spontaneously and has a delta G less than zero. If you do have a potential on the gradient and your molecule has a plus or negative charge on it, a full plus or negative charge, then if the concentration gradient, the effects of the concentration gradient on that molecule and or that ion, and the effects of the potential gradient on that molecule or ion is less than zero, then that type of movement will be diffusion. Okay, so we talked about this at the end of the last video, and here's a table showing you the concentration of ions across a cell membrane. Now, these concentrations, these concentration gradients, will not change as long as the cell is alive. And we're talking about cells in a multicellular organism, and this is basically humans, typical mammalian cells, as being said here. So in typical mammalian cells, these gradients will not change. Now, so far in this class, we've talked about two examples of structures being formed, and that was the phospholipid bilayer and protein folding, and then proteins, subunits coming together to make bigger structures, and saying that those things happen spontaneously, meaning that there's no need for input of energy. They will happen spontaneously, and that really has to do with the fact that entropy is being increased because ice-like cages around hydrophobic surfaces are minimized, right? And the one hint that we got that, okay, well, where does all the calories go? Like, why is the cell producing so much ATP? What does it need to use that ATP for? Where is the structure that requires ATP, meaning that you're moving the system away from high entropy to low entropy? We got an example of that with the asymmetry on the cell membrane with its phospholipids, right? Phosphatidylserine is always on the inner leaflet. Glycolipids are always on the external leaflet, and phosphatidylinositol is always on the inner leaflet, and that is structure that doesn't happen spontaneously. That there's a requirement for energy to be spent to move the phospholipids on the correct leaflet. Now here is, again, gradients that are far away. Some of them are very far away from equilibrium. And the equilibrium we're talking about is from the equation above here that we just talked about previously. This equation and the calculations that we sort of did here and the equation that we have here. Right? So, many of these ions that we're showing here, they're away from equilibrium. And the questions that you have in the homework in the first two questions, it's asking you to calculate what the equilibrium potential for these ions at these concentrations are. Because the resting membrane potential is usually around minus 50, minus 70 millivolts. So assume that question is asking you, assume that the cell's resting potential, membrane potential, is minus 70 millivolts, and then calculate the equilibrium potential for each one of these ions at these sort of concentrations. And when you do that, you'll find that some of these guys are really far away from the membrane potential that is needed for them to be at equilibrium at these concentrations. Now, why are we worried about the membrane potential and not about these gradients, these concentration gradients? Well, these gradients never change. These are kept this way at these concentrations across the cell membrane. And a lot of energy is spent by your cells, in some cells up to 2/3 of the ATP produced, that is directly or indirectly powering creating these gradients. Now, in the case of sodium, you have 5 to 15 in millimolarities and 145 millimolarities outside. Potassium is almost the opposite, right? So this is sodium, this is potassium. Magnesium, 2+, a lot more outside than inside, about 5 times more or 4 times more. Now, calcium is one of the steepest gradients right here. And I want you to take a look at calcium. Right here, right? 10 to the minus 4 within the cell, within the cytosol. And this should be really called the cytosol, not the cytoplasmic concentration. So 10 to the minus 4 in the cytosol and 1 to 2 millimolarities outside. Now that's a 10,000 times difference between internal concentration and external concentration. That is a huge concentration gradient. For sodium, 10, right? That's it, 10 times. So there's a reason for this. And it's something that we're going to be talking about a little bit in this section, and something we're definitely going to be touching upon in section 3. Anytime calcium concentrations increase a little bit within the cell, within the cytosol, there is some sort of response by the cell. In this section, we're going to be talking about muscle contraction. Calcium concentrations go up in a myocyte, a muscle cell. You get contraction of that cell, for example. So And H plus, of course, to maintain pH. And Cl minus, again, about the similar gradient as sodium, except Cl minus is negatively charged, so its potential gradient will be pointing in the opposite direction as sodium. So that is of some importance. The two that are really interesting to us for this section that we're going to be talking about a lot, the two ions, are sodium and potassium. Now you might be asking for yourself, do I have to know all this? Yes, you do. You will have to know all of this to understand what's going to be happening later in this section. So appending our definition of channels and transporters. Channels move their molecule or ion down its electrochemical gradient. So this first one is something for channels. This one is for transporters. There are two types of transporters. Transporters that mediate passive movement of their molecule down its electrochemical gradient, aka diffusion. Transporters that move solute against its electrochemical gradient in a process called active transport. Okay, so the picture here is kind of showing you that. You have simple diffusion, which doesn't require any protein involved channel or transporter. For example, O2, CO2. Those molecules will easily diffuse across a cell membrane. You have a channel here, which is facilitating diffusion of its molecule or ion down its electrochemical gradient. Then you have transporters. So this first one right here, this is just a normal transporter that's moving a single molecule or ion down its electrochemical gradient, facilitating its diffusion. And then you have this one right here, which is using some sort of source of energy, right? It's being coupled to, for example, ATP hydrolysis or something else, where a molecule is being moved against its electrochemical gradient. If the molecule does not have a charge, then it's moving it up its concentration gradient. So first we're going to talk about transporters. Transporters. A transporter protein in a cell membrane has three conformations or native states. And we have talked about proteins that have more than one native state. Open to the extracellular side, where the solute binding pocket is exposed to the extracellular solution. The occluded state, where the transporter solute binding pocket is not exposed to the solution on either side of the membrane. Open to the cytosolic side, where the cell binding pocket is exposed to the cytosolic solution. The stability of each state is equivalent, but a transporter must go through the occluded state in order to change which side its solute binding pocket is exposed to. A transporter which facilitates diffusion can go to the occluded state with its binding site empty. So, a lot of things here. Let's look at this picture. So what you have is you have three states. You have the occluded state, which is the state where the binding pocket is not exposed to either the outside or the inside solution, right? You have an inward open, where the binding pocket is exposed to the inside solution, and you have an outward open, where the binding pocket is exposed to the external solution, or the outside solution. Now in this case, we have a molecule, and we're only worried about the concentration gradient, because this molecule does not have a charge on it, right? So no need to worry about the potential on the membrane. No charge. It's like the potential doesn't even exist. Right? So we're not even worried about that. Now, what these arrows are telling you here and here is this protein can flip from inward open to occluded and actually go from occluded to inward open again, right? There's no rule saying once it goes from inward open to occluded, it must go to outward open. Same way from outward open. It could go from outward open to occluded and go back to outward open. There's no rule saying that it must go to inward open after going to the occluded state. So this is completely probabilistic. it will just happen by itself. The three states are about the same energy level, so any of the three states can happen. It's just that if you want to go from inward open to outward open, you must go through the occluded state. This sort of trajectory in this direction or this direction must occur. Now, the transporter, in this case a uniporter, which we'll define later, meaning that it only binds one molecule at a time, can go to the occluded state completely empty-handed. In this picture, it's not going empty-handed, but it could simply be not picking up anything here and go to the occluded state empty-handed, and then go from the occluded state back to inward open or go to outward open. There is no rule saying that it must necessarily pick up something to switch from one state to another, or at least from inward open to occluded or from outward open to occluded. It could go empty-handed. And now the question becomes, wait a second, if this is all probabilistic and this can do all these things and there's no guarantee that you go in one direction or the other, how does this facilitate diffusion? And that is the question that we're going to answer in the next video. Okay guys, thank you. Bye-bye.